Well, hello. Good morning, afternoon, evening, wherever you happen to be. Thanks very much for tuning into this talk on the GnRH neuron pulse generator in vivo. And thank you to the Endocrine Society for the opportunity to present this work. My name's Allan Herbison, and I've recently just moved from the University of Otago to the UK, to the University of Cambridge. What I want to do today is talk around a couple of recent papers that we've been involved with that have come out that I think shine some light on how the GnRH pulse generator is operating. I have nothing to disclose. This slide is really just a mere formality for this audience, but just reminds us that pulsatile GnRH secretion is what males do all the time, and that females have pulses as well as this surge pattern here, and that these pulses are really important for fertility. Here's normal LH pulses in a woman, and here's in PCOS, and here's in hypothalamic amenorrhea, both conditions associated with infertility. So we're using rodent models to try and understand these modes of GnRH secretion. I think it's worthwhile perhaps just to go over a quick overview of what the GnRH neuron looks like in the mouse and the rat. This is a side-on view. Here's your GnRH neuron cell body scattered out in the basal forebrain, and here are their projections going all the way down to the median eminence, where we're releasing GnRH into the portal circulation. Now, we do know quite a lot about these GnRH neurons. For example, we know that they initiate the action potentials out here on their proximal dendrites. And we also know a bit about their synaptic innervation patterns. Relatively few inputs onto the cell body itself, but many clustered out here onto these proximal dendrites. And then, interesting, unusually, and possibly even uniquely in the brain, we have a very large number of synaptic inputs down here onto this projection here. Indeed, the highest density of synaptic inputs on the GnRH neuron is down here. So these rather unusual projections then are coming from both ends of the GnRH neuron, and they're transmitting action potentials down to the median eminence, so they're operating as axons, but they're also receiving synaptic input, which means they're dendritic-like. So we call these projections, because of their blended dendrite and axon functions, we call them the GnRH neuron dendron. What this means then is that the GnRH neuron has two sites at which it can be regulated, at the classical way up here at the cell body, and then another mechanism down here at these dendrons. And we've been interested for some time in working out how that actually operates. And what I want to do today is just talk briefly about some work that we've been doing with a really nice collaboration with Dr. Li Wang and Professor Yanping Quan at the 9th People's Hospital in Shanghai. So we decided that the thing to do would be to try and inhibit the activity of the GnRH neurons at this location or this location to try and get an idea about what those parts of the GnRH neuron are doing. So to do that, we use chemogenetics, the DREADS, pretty standard now, using HM4D-GI. And the idea here was to put HM4DI into all the GnRH neurons that were projecting to the median eminence. So that involved an AAV retrograde virus. Li Wang and her colleagues injected into the median eminence here, and this infected all of the GnRH neurons projecting this area. It turned out to be about 65% of the GnRH neurons. And then at a later time, they came back, and now they injected the DREAD activator CNO 1 microliter bilaterally into the medial basal hypothalamus and looked to see what effect that had both on the surge and on LH pulses. And here's just a picture of those dendrons just as they're heading down into the median eminence, and here's just showing you the M-cherry tag on this HM4D-GI in these GnRH terminals and dendrons. So here's the LH surge. So this is using an overrectomized estrogen plus progesterone model. And here this shows you actually the LH values of all the animals in this particular study. We decided to take blood samples here at this point here, and then 19, 20, and 21, so as to not take blood samples around this critical point when we know the LH surge is actually initiating, but we don't want to disturb the mice beyond what is required. So perhaps you get these sort of unusual lines here. This doesn't mean that the surge started at 1600, it's just connecting the two time points between those samples. But nevertheless, you can see very decent surges going on here in all these animals when saline was injected into this area. However, when you put in the DREAD activator, you can see the L8 surge is completely abolished. Similarly, if you look at pulsatile L8 secretion, now this is an overactimized female mice, and here's some nice normal LH pulses with saline. And when you put in CNO, there's a dramatic reduction in LH. You still get these tiny little blips that are probably pulses going along here. The amplitude of pulses is vastly reduced and the frequency. So you do get quite a substantial reduction in pulsatile LH secretion as well. And again, that's probably not that surprising. The CNO here is activating the dendrons as well as the terminals, so it's inhibiting the secretory activity of the GnRH neurons, so we get these effects on the surge and the pulse. Key experiment, however, was here when Li Wang and her colleagues injected CNO now into the preoptic area. And these are quite decent volumes, 2 microliters bilaterally into the preoptic area to try and impact as many GnRH neurons as possible. Here's the L8 surge profiles. Here's with the CNO. Here's the mean data. You can see that again, the L8 surge is completely abolished. So indeed, we need our GnRH neuron cell bodies to generate the surge. However, intriguingly, pulsatile L8 secretion continued on completely normally, and here's the mean data here. So this is a little bit confronting because it suggests that the GnRH neuron cell bodies are really not involved in pulsatile L8 secretion. So we were aware of that, so we decided to also use another methodology to inhibit these cells. So we used inhibitory optogenetics, in this case, archirhodopsin. So this was a mouse model in which, genetic mouse model in which archirhodopsin was put into all GnRH neurons, and then bilateral optic fibers put in where we can shine light to inhibit the activity of the GnRH neurons. So here's the L8 surge experiment. In the black shows you the L8 surge mean levels in animals that had the injection but no laser. The blue shows you what happened actually when we put blue light in. So blue light will not have any effect on archirhodopsin, but it's a very important control when you're shining light into the brain of an animal. And the red light here shows you what happens when we actually, when we actually activate the archirhodopsin. And you can see the surge is completely abolished. So as we found here. However, when we then went and looked at pulses, and now we've shown yellow light in for this 40-minute period along here continuously, you can see once again it had absolutely no effect on pulsatile L8 secretion. So these data suggest then that the GnRH neuron cell bodies are really not involved in the mechanism of pulsatile L8 secretion. This is quite compatible with this model here, in which we have the GnRH neurons essentially as not much more than slaves, sadly, to 2 different pulse generators. So we have a surge pulse generator here, innervating the GnRH neuron cell bodies and proximal dendrites, and that's generating the surge once every cycle. And at the other end, we have the pulse generator, the kisspeptin neurons, and we'll come to them in a minute. The pulse generator acting on the dendron down here to generate all of these individual pulses. So what about this dendron area down here? It seems pretty important. We know that the arcuate kisspeptin neurons are the pulse generator, and we know that these cells project down into this area. So we were very interested to look at actually how that signaling occurs. What is the fundamental mechanisms going on there? It's always good to start with some anatomy. So here's some work this was done by Michel Hurd in the lab. And what Michel did was he wanted to look at what is the relationship of those Kindi neuron projections down into the dendron. And here he did staining for kisspeptin and neurokinin B. So here's a purple kisspeptin fiber here. And what he found was that each kisspeptin fiber wound its way through and around all those GnRH neuron dendrons. And he found that on average, each kisspeptin fiber impacted or made close associations with about 4 or 5 dendrons. So this generates what you would call a divergent mechanism of transmission, with one kisspeptin neuron influencing multiple GnRH neuron dendrons. So we looked here, and you can see that these look like close appositions, but of course you can't tell with regular confocal imaging that this is whether this is a synapse or not. So Xiao Yo, starting with me, she'll start up expansion microscopy in the lab, which is a really nice, simple method which actually blows up your tissue specimen so that you can use regular confocal imaging, but now you're able to determine synapses. So here's an individual image that Xiao made. Here's the dendron, again in green. The kisspeptin staining is in pink. And now we have synaptophysin in orange or gold. So what we're looking at is those locations where kisspeptin is being released from these kisspeptin fibers as they wind their way through the dendrons. So these things look like they have a very close association, which is, as we saw before. But if you do a line scan through here, and this is the line scan, this here shows you the dendron, and here is the kisspeptin bouton with the synaptophysin in it. There's actually very, very little overlap here. Now, we've done quite a lot of work with expansion microscopy on the dendron and on the GnRH neuron cell body, and we know that a bona fide synapse actually requires an overlap, a very substantial overlap in this signal. So this suggests that there is not a synaptic apposition here, and indeed when Xiao went in and she looked at many, and in fact every single one she looked at, she saw that there was no actual synaptic association between the kisspeptin and the dendron. Here's another example down here. Here is your kisspeptin fiber. There's actually some synaptophysin in there as well. Here's the dendron, and here's an unidentified terminal with synaptophysin just sitting on the other side of the dendron. So when you line scan that, here's the kisspeptin synaptophysin sitting there, no overlap onto the dendron. But here's this other synaptic terminal here, which has a significant overlap. This here would be representing a synapse. So this mechanism of transmission is called volume transmission, and the concept is that kisspeptin is basically being sprayed out from these little boutons around the dendrons, but not specifically at any specific synaptic site. So kisspeptin neurons, or the Kindi neurons, as we know, they've got kisspeptin, dynorphin, NKB, and glutamate in them. We know that from making recordings of these cell bodies that they actually express NKB. Here's a recording of that Simon de Croft made many years ago now, showing NKB activating kisspeptin neuron. Here's dynorphin inhibiting that cell. And interestingly, kisspeptin has no effect whatsoever. So the receptor here, the complement here is probably something like this. There's no kisspeptin receptor, but there's this signaling going on, and it may well be that this is involved in the synchronization of these cells. So what's going on at this end of the Kindi neuron? And for this, we might reflect a bit on Dale's principle that actually was formulated by Eccles. And the principle is simply that a neuron releases all of its neurotransmitters at each of its synaptic appositions. So whatever it's sending to this synapse, it's also sending down here. So that would suggest then that all of these neurotransmitters are potentially operating on the dendron. So how do we look at that? The dendrons are tiny, tiny little structures. You can't measure the electrical activity of them, but you can use GCamp imaging to measure calcium, intracellular calcium in them, which is a good proxy for electrical activity. So here we're using a GnRH Cree mouse, and we're putting a GCamp6, the calcium indicator, just into the GnRH neurons. And we're using a thing called the French cut, which was developed by Stephanie Constantine when she was in the lab. And essentially what you do is you flip the brain up and you take one single cut from the base of the brain. And here we are looking down on it. Here's the median eminence of the GnRH fibers. And these little dots are the individual GnRH neurons sitting out here in the anterior hypothalamic area. If you go in here, and now you need a confocal to look at that, you can see all the individual GnRH neuron dendrons as they're coming down, moving into the median eminence. So you can do that, and you can actually puff things on here. And here's a little video. This is what Carl Eimanger did, and he's puffed kisspeptin on, and pretty soon you'll see a whole lot of GnRH neuron dendrons responding with increased calcium to that puff of kisspeptin. So that was done in the mouse. Recently, Sinwai Liu has been able to use exactly the same approach in the rat and found the same thing. Here's just some individual traces of dendrons showing kisspeptin activating the rat GnRH dendron as well. So what about all those other neurotransmitters there? Well, Sin-Wai Liu went on and looked at this. Here's your kisspeptin response. We then decided to look at glutamate, and we were amazed to find that glutamate, AMPA, and NMDA actually had no effect whatsoever on these dendrons. We moved on to NKB, and again, NKB had no effect. This is the control. There's often a small 3 or 4% increase or change in calcium with the control ICSF, and here's the NKB, no effect. However, if we switch animals and make it a kisspeptin GCAMP animal, and now we do that French cut, flip it up the other way, we can now look down on the kisspeptin neuron cell bodies, and you can puff on NKB, and you can see it's very potently activating these cells. So it's not a technical problem. And again, dynorphin, absolutely no effect. And being inhibitory, we thought, well, maybe it acts on the kisspeptin activation, but again, absolutely nothing. So we can take this picture, and it looks very much like, actually, even though there's all these neurotransmitters here, and actually they are all being released, and I don't have time to show you that data, but you can look here in the recent paper. But the only neurotransmitter they're taking any attention of is kisspeptin through the GPR54. So this provides a really quite intriguing situation, actually. You have what I'm calling converse patterns of co-transmission. Whereas down here, we're getting a release, and the only thing that's being listened to is kisspeptin. Up here, you have all these things being released, and it's listening to everything except kisspeptin. So we were wanting to take this in vitro data and try and look at whether we could see what might be happening in vivo. And we rationalized that if we were to look at this system, but look at it in an animal in which kisspeptin was deleted, so if we take kisspeptin out, then presumably it would have no effect on these synchronization events because kisspeptin is not active here. But down here, the removal of kisspeptin would mean that there would be no signaling between these two elements, and it would abolish pulsatile LH secretion. So luckily we're able to do that. We can use GCAMP fiber photometry. This is a methodology that allows us to look at the synchronized activity of these kisspeptin neurons as the mouse walks around its cage. So here, for example, this is in an overactinized female mouse. You can see here, these are these synchronization events. They last about 1 to 2 minutes in duration. And here they are occurring over a 2-hour period. So each one of these is a period when the kisspeptin neurons are all firing together. At the same time, if we take blood from the tail tip of these animals, we can look at LH, and you can see here there is, as you always, a perfect correlation between these events and LH. So the neurons synchronize, they drive release of GnRH, and you can measure a pulse of LH 3 to 4 minutes later. So what happens then if we do this in that model that I was talking about that has no kisspeptin? Well, here, really, as we suggested, it has no impact. These synchronization events just carry on completely like normal in the absence of kisspeptin. But if we take LH from the tail tip, there's absolutely zero LH pulses. So in these animals, the poor kisspeptin neurons here are synchronizing, they're trying to generate pulses, but essentially they're firing blanks down here because they're no longer able to release kisspeptin, which is what's required for the dendron to generate the pulsatile LH secretion. So just in summary then, this is the diagram of the pulse generator, and you'll note that it lacks the GnRH neuron cell bodies because we don't think they're actually important or required for pulsatile LH secretion. I've shown you that these kisspeptin neurons are using divergent volume transmission. These little, the way I've indicated here, with these little synaptic boutons winding their way through the dendron. Kisspeptin neurons are using quite different and converse patterns of neurotransmitter receptor expression at these 2 points. And it's at this point here, in fact, the only thing that's important is kisspeptin. It's the only thing that the dendron is actually listening to. So I think these studies provide some further steps forward in understanding the pulse generator. These are the 3 papers here that I've referred to in more detail. Maybe of interest to read those. And finally, just to mention those people that have done the work. This is one of the pictures Well, the last pictures taken of the lab in New Zealand, the remaining people, here's Xin Liu, and Rob Porteous and Isaiah Chong and Jamie McQuillan that were involved in the photometry studies. Here's Jenny Clarkson, who was supported by the HLC and Marsden Fund. And here on the other side of the world, of course, banisters go in the opposite direction. This was the beginning people of the new lab in Cambridge, and here's Xiao Yo holding a 3D model of a GnRH neuron. And here we're supported by the Wellcome Trust. Finally, just to mention the really great collaboration we've had with Li Wang and Yangping Quang in Shanghai, Bill College from ICE, and Carl Einmunger for help with the Dendron work. So that's it. Obviously, there's no questions, but here's my email address here, and I'll be very happy to entertain any questions, thoughts. So thank you very much, and I will sign off now. I can.